Hi guys, we are back again today and we are going to wrap up our study of macromolecules by talking about the last macromolecule, which is the nucleic acid. But before we jump into that, I want to make sure that you have reviewed the proteins. So here's a few different questions that you could ask yourself to say, do I know this? Okay. Keep in mind that these questions here are very basic, like they're very just memory. I can remember that. I know we talked about that. I know what that is type questions. AP test questions are not like that. You have to apply knowledge to a different situation. So if you're not getting these questions without having to look up notes and look up something, you definitely do not know it well enough to be able to apply it to an AP question. Okay, so keep that in mind as we continue to go through. If you have questions about something that we've covered and you're still unclear on it, then let me know and I'm happy to help. So let's jump into the nucleic acids. So nucleic acids, an example of a nucleic acid would be DNA and RNA. DNA is deoxyribonucleic acid and RNA is ribonucleic acid. Now, you're probably familiar with DNA. You know that it's your genetic material, that it makes you. If you know even a little bit more, you might know that it lives in the nucleus and that you get half of it from your mom and half of it from your dad. And that it's this twisted spiral staircase that's called a double helix. And that's kind of the structure of the DNA, okay? You may not be as familiar with RNA. So RNA, there's several different types, like 100 different types. We'll focus on 3 types, but we actually won't get to that today. So we are going to cover just the very basics today, but we will do a whole unit on DNA and RNA later on, like after Christmas. So, but let's try to get the basics of nucleic acids today. So nucleic acids have genes, okay? Genes code for proteins, okay? I'm going to write that up here because I think that's important. Nucleic acids are made up of genes, which code for proteins. Okay? And so that's kind of how we tie in our nucleic acids that we're talking about today with our proteins that we talked about previously. So what is a gene? A gene is just a short segment of DNA. It's just a piece of the DNA. So remember, the DNA is a huge molecule, and it codes for lots and lots and lots and lots of proteins. And so if you just take a small segment of that codes for one specific protein, that is a gene, okay? That's what a gene is. And so genes are only in DNA. Genes are not in RNA, but the RNA is really important in the process of making a protein. So what we've got here is we've got DNA that lives inside this nucleus, okay? The DNA, we want to keep it secure. We want to keep it under lock and key. We do not want to send it out into the cytoplasm of the cell because in the cytoplasm of the cell there are enzymes. And one thing that enzymes can do, aside from speed up chemical reactions, is enzymes are known for cutting things up. They cut the DNA up. They degrade the DNA. But we don't want to lose our DNA because it's our genetic material and it's really important. And so instead, we have this other molecule called the RNA. And specifically, the one we're going to talk about here is the messenger RNA. So the job of the messenger RNA, it goes back and forth between the DNA and the ribosome. The ribosome is where the proteins are made, at the ribosome. So we have to go to the DNA and get the instructions and copy it down and take it to the ribosome before we can make that protein. And so that is the job of the mRNA, the messenger RNA. So the messenger RNA is just single-stranded, okay? You see it doesn't have that double helix look. Sometimes it's referred to as half of a helix. But otherwise it's pretty similar. There's really like 3 main differences. One is the double versus single-stranded. The next is the sugar. So in DNA is deoxyribonucleic acid because it has the sugar deoxyribose. Remember, oses are sugars. RNA is ribonucleic acid because it has the sugar ribose. Okay, so differences are the single versus double-stranded, and then the sugar that is involved, okay? And then a third difference are the nitrogen bases, and we'll talk more about those in just a moment. So, like we just said, DNA and RNA together direct protein synthesis, which happens at the ribosome. So, nucleic acids, again, they are the polymer, they are the big molecule, and they are made up of monomers. And so the monomers of nucleic acids, there's actually 3 parts of a nucleic acid, or 3 parts of a nucleotide. They are the 5-carbon sugar, which would be the deoxyribose here, or the ribose in RNA. The phosphate group, which we've seen those before, and then the nitrogen base, okay? So you see all 3 of these components here. Phosphate group, nitrogen base, and the 5-carbon sugar, okay? And again, if you remember our sugar structure, we've got these 5 points to indicate the 5 carbons, and so we would call that a pentose sugar because it has 5, pent is 5, and then ose because it's a sugar. Okay, so the 5-carbon sugar, the phosphate group, and the nitrogen base. You gotta know that. 3 parts of a nucleotide: 5-carbon sugar, phosphate group, nitrogen base. You need to stay in your sleep. So that is the monomers. There's not just one monomer like in proteins. We say the monomer is a nucleic acid, amino acid. In a nucleic acid, there are 3 kind of monomers. So nucleic acids are made up of 5-carbon sugar, phosphate group, and a nitrogen base. So, or you could, even more simply, I guess you could say nucleic acids are made up of nucleotides. This is Miss Brown. Hey, Barbara. Yes. If you can, yes. Okay, I'll see you soon. Bye. The 3 parts of the sorry, I don't know where I was. I got a phone call. So, more simply, you might say that a nucleic acid is made up of nucleotides, and then nucleotides are made up of a 5-carbon sugar, a phosphate group, and a nitrogen base. Okay, so that is probably the most accurate depiction, but it's important to know all 3 of those steps. So I'm going to skip over to here and show you. So this is demonstrating RNA. Okay, this diagram is RNA. And so you see it's half of a helix. It doesn't have the other half of the ladder. So this is a nucleotide. You've got the 5-carbon sugar, the phosphate group, and the nitrogen base. 5-carbon sugar, phosphate group, nitrogen base. Okay, so in this one, it's just showing simply 4 nucleotides to make up this whole thing, which is the nucleic acid. Okay, and those are held together, while I'm on this slide, by phosphodiester linkages. So the bond that holds the sugar and the phosphate, so our sugar and our phosphate, these bonds here that hold those together are called phosphodiester linkages. Okay, now in DNA we have a few more bonds that would hold the bases here together. So if you looked at DNA, the other side of this picture would be basically a mirror image. And so this bond is a hydrogen bond. And we'll talk more about that again later. But the reason that this is important, this phosphodiester bond, or phosphodiester linkage, is a covalent bond. So it's strong, okay? It's holding these molecules together. and it makes up the sugar phosphate backbone. That's what we call it in the DNA and the RNA, the sugar phosphate backbone. If you look down through here, on the sides you have sugar phosphate, green and yellow all the way up, okay? And then in the middle you have your bases, okay? And then if you had it on the other side, you'd have another base connected to another sugar and another phosphate. Okay, here's a picture that's better. So you have the sugar phosphate backbone, okay? Sugar phosphate backbone. You've got the bases in the middle, and then the sugar phosphate backbone on the other side with the hydrogen bonds here. Now, we remember about hydrogen bonds. They are an intermolecular force between two different molecules, and so they're relatively weak, okay? And that's important because when the DNA replicates, which happens every single time that you make new cells and you copy your cells, the DNA has to be copied and replicated. This has to break in half to be able to copy it. And so we want weak bonds here that can be broken in half easily, but also be put back together easily. Okay, so these are your phosphodiester linkages, and then these are your hydrogen bonds in the middle. Okay, one other thing that you may notice about the sugar phosphate backbone in these pictures is that this one, the sugars are like pointing up, okay? This one, the sugars are pointing down. We will talk about this again when we get into DNA, but this is referred to as the anti-parallel nature of DNA. The strands run opposite one another, okay? I'm going to go back before I get too far ahead of myself. So, in the DNA, again, we are made up of nucleotides, which have a 5-carbon sugar, a phosphate group, and a nitrogen base. So, in DNA, DNA has 4 nitrogen bases, and then RNA has 4 nitrogen bases, okay? The 4 nitrogen bases in DNA are adenine, thymine, guanine, and cytosine. RNA is a little bit different though. It does not have thymine. Instead, RNA contains uracil. So adenine, uracil, guanine, and cytosine. And you see that over here as well. We've got the thymine in DNA, the uracil in RNA. And usually those are just abbreviated with a letter. These are the nitrogen bases. So these nitrogen bases are going to fall in one of 2 classes. Nitrogen bases are categorized as either a purine or a pyrimidine. The purines have 2 ring structures, like you see up here, okay? Adenine and guanine both have that double ring structure. The pyrimidines have a single ring structure, like cytosine and thymine, and also uracil is a pyrimidine. Okay, you see the single ring here in uracil. Now, what's important is that a purine always pairs with a pyrimidine. And that's really important to make sure our steps in the ladder are equal widths. If you had A and G paired together, your step would be like 4 rings wide. And if you had C and T paired together, the middle of your step would be like 2 rings wide. And so that just doesn't work to have those steps that are different widths, because the backbone would be like curvy, so to speak. So what we know is that the bases always pair in this way, and you gotta know this, and you gotta know it by its name. Chargaff's rule tells us that A pairs with T and G pairs with C. Okay, in DNA, A pairs with T and G pairs with C. In RNA, A pairs with U. So if I give you a sample sequence of DNA, you should be able to write the other half of the helix, the other side of the DNA, or the RNA that it would make up if it was being copied into RNA to be able to make like a protein. So let's just do that here quickly. So if we are gonna, if one side of our DNA is this strand that I've written, the other side should be T A A T C G T A C G T. Okay? Just using the A pairs with T, G pairs with C rule. Okay? Now, if I want to take this half of the DNA and make a strand of RNA, let's look and see what that RNA sequence would be. T is going to pair with A. A is going to pair with U. U, A, G, C, A, U, G, C, A. So what you notice between this strand and this strand is that everything is the same except the T's are replaced with U's. Okay? So that's how the base pairing rule works. Okay? And I don't think I covered this statement really, but it's talking about the anti-parallel direction, which is what I just described here. Okay? How the two strands run in opposite directions in the spiral DNA. Now, that doesn't hold true for RNA because in RNA there's only one strand. So, 3 differences in DNA and RNA that we were just talking about a moment ago. The sugar, so we got deoxyribose versus ribose. The bases, we've got A, T, G, and C in DNA, and then we've got A, C, G, and U in RNA. And then, the single strand in RNA, the double strand in DNA. Those are the 3 main differences between DNA and RNA, which are our nucleic acids. Okay? I think that wraps everything up today. I think we hit it all. Again, go back through this. Try to grasp as much of it as you can. We'll go over it again next week when you all are here with me. And I think you have an assignment to do on this today. You've got a biological molecules worksheet that just kind of reviews all of the different macromolecules. So complete that and let me know if you have any questions. You all have a great day.